All right, welcome everyone. In just a moment, I'm going to ask you to grab off the table. Also, it is linked via Canvas as a PDF if you are joining us on the video today. A worksheet that I call "Can You Draw Mitosis?" On December 21st and or 22nd, you will be taking your biology test on cell cycle, mitosis, and meiosis. And on Monday or Tuesday, we drew step by step the processes of mitosis. And I said on your test on the 21st or 22nd, I will ask you to draw step by step and identify the key processes of mitosis. I also told you on Monday or Tuesday that in class on Wednesday or Thursday, today, I will ask you to draw this from memory. So that's what we're going to do on this. And once you get it, I'll kind of explain, I'll show this to you now so you should be able to go. There's a you draw, we draw. I'm going to ask you to do the column of you draw. In the first column, it's the phases or the stages of mitosis. We're going to start you with interphase, and then we're going to go into something and something. Remember, our puppy mitosis pees all over the floor, so she needs something to help us, so we clean it, keep the mess. But these very last two slides, or these very last two boxes, I want these last two boxes, I'm going to tell you that we are going to end with interphase. Is what we want to end with. So we're going to start with interphase, we're going to end with interphase, and that's what I want you to draw. I have some colored pencils sitting right next to it. I would encourage you to grab colored pencils to draw it like we did in class. That's going to be the same colors. We did 4 chromosomes in class. I'm going to draw four chromosomes on the screen. You could do 6 chromosomes, you could do 8 chromosomes, you could do 46 chromosomes if you really wanted to. Don't recommend it, but you need pairs. Okay, because we start with the diploid cell, complete set of genetic information. So I want you to grab a piece of paper, colored pencils are there, and draw from memory, not from your notes. I want you to pull from memory. I want you to struggle through this. I want you to get down what you can. And I'm going to give you about 5 minutes, and we'll kind of see where we're at from there. Okay, so go ahead and grab that sheet, try it from memory. You've been given some time now to go through and draw these steps. On your test on December 21st or 22nd, I expect you to be able to do this from memory without any resources besides the one between your ears. We're going to go through and you're going to check this yourself. You drew it here, we are going to draw it here. Now it's kind of up to you. What do you think is best for your learning? Some people are choosing to draw it 100% step by step over again. Some people are choosing to go through and they're checking that, and if their box is wrong, they're like, oh, and they're crossing theirs out, and then they're drawing the correct one over here. That's kind of a nice option. I think drawing step by step is good. But by physically crossing off the ones you got wrong is telling you, like, hey, I did this one wrong. I screwed up. I need to work on this one a little bit. And your brain is kind of telling you that. So you do what you want, what you think is best. At the end of the period, you will have a 100% complete, accurate drawing of this. So we start with interphase. I'm going to talk about the key steps. We aren't actually going to draw each and every step, but during the process of interphase, we have a cell that has a nucleus, and in that cell is some genetic information. That's a bowl of uncoiled spaghetti in the form of chromatin. And during interphase, there's 3 phases of interphase, expect you to know. G1, S, G2. During G1, the cell grows. During S phase, the DNA replicates. During G2, the cell prepares for division. It does its day-to-day stuff. Muscle cells do muscle cell stuff. Bone cells do bone cell stuff. Okay? Our puppy mitosis is just a puppy, so she needs a mat, okay, a PMAT, or pass me a taco. When we talk about passing me a taco or PMAT, the first phase is referred to as prophase. And during prophase, we have a cell. During prophase, that nucleus that we had dissolves, is chopped up, is broken down, it disappears. That's what those dashed lines are representing. We get these things called spindle fibers that are starting to form in the cell. Now, technically, they aren't opposite ends of poles yet, but whatever. we have these spindle fibers, we have these ropes that are gonna pull genetic information around, and that genetic information is in the form of chromosomes. And I am going to draw, oops, those should have been a little bit more size appropriate. Look at this, they are much closer in size now. Okay, so we have two pairs of chromosomes. Where I drew them is completely random, it doesn't really matter, but I have a pair of big chromosomes, I have a pair of little chromosomes. These are homologous pairs, these are homologous pairs. These have the same genes, these have the same genes. One of these came from dad, one of these came from mom, one of these came from mom, one of these came from dad. These are called diploid cells. There's a pair of chromosome number 1. There's a pair of chromosome number 2. They're in chromosome form. Pass me a taco or PMAT. The M stands for metaphase. Metaphase. And what's the other M word that goes along with metaphase? And remember, we are, middle is correct. Middle, meta, middle, middle, meta, middle, meta. Meta, middle, the middle is what goes to the middle. Well, mitosis is all about dividing genetic information. What goes to the middle? The genetic information goes to the middle. So we have our cell, and in the middle of our cell, we get some all of the genetic information lines up in the middle. Now, the order in which I drew these down the middle is random and doesn't matter. I chose to go big red, little red. Random, it doesn't matter. These could be flipped. This one could be in between these. It makes no difference. It's random, it doesn't matter, it's random. Why the heck did those things go to the location they did? Because of those darn spindle fibers, those lassos, those ropes are pulling on the chromosomes to align them where they are going. Those spindle fibers pulled them along the way, and they pulled them to the middle. The genetic information went to the middle. Meta, middle, chromosomes go to the middle. We get on the front before I flip it to the back? Okay, meta, middle. Pass me a taco or PMAT at, okay? The A stands for, I changed colors, that's actually, oops, I had written, whatever. Anaphase. Anaphase, and what's the other A word that goes with anaphase?
Apart.
Apart, or you could say away, either one, but apart. So during anaphase, something is pulled apart. Now remember, mitosis is all about genetic information. So we are talking about the genetic information being pulled apart. Now I am going to flip it over to the front, just because I want to look. I went big red, green, little red. I want to keep that order just to be consistent is why I was looking. So my chromosomes are pulled apart. My big red chromosome gets pulled apart. My big green chromosome, oops, that was really big, gets pulled apart. My little green chromosome gets pulled apart. And my little red chromosome gets pulled apart. What do we call these things? These two things that I'm pointing to?
Sister chromatids.
They're sister chromatids, yeah. So this chromosome gets pulled apart into two sister chromatids. And what's true about sister chromatids? Genetically, they are identical to one another. Very good. Okay, genetically, they are identical to another. Why did they get pulled apart? Because those spindle fibers pulled them apart. PMAT, or pass me a taco. The T stands for telophase, and I'm going to slash it with cytokinesis. So when we talk about cytokinesis, what's going to happen is we are going to pinch this cell right here. We're going to pinch it. So when we draw that, because that's what cytokinesis is, the pinching of the cell membrane or the pinching of the cytoplasm, I often see I'm pinching this right here and I'm pinching it and pushing that in, that's why it looks like that. Now during telophase, we're going back to interphase. So basically everything that happened in from interphase to prophase has to be undone. So in interphase to prophase, the nucleus dissolved. Now what we have to do is we have to replace or rebuild, restore the nucleus. Now these are each going to be their own separate cells, so therefore they're each going to have a nucleus that goes along with it. Our chromatin is going to go to our chromosomes are going to go to chromatid. I don't want to draw it quite like that yet. I want to still draw it in chromosome form. And I'm drawing these in random spots. It makes absolutely no difference where I draw it. Now the reason I'm drawing them like this still is they will uncoil, and we'll show that back in interphase, which is what we're going to end up with. But if we count how many, this is identical to this is a sister chromatid to this is a sister chromatid to this is they're genetically identical, so this has the exact same genetic information as this, because there's 4 sister chromatids that are genetically identical to these 4 sister chromatids. But if I count the number of chromosomes, we could say 1, 2, 3, 4. There are 4 chromosomes on this cell. There are 4 chromosomes in this cell. There are also 4 chromosomes in this cell. There's 2 big ones, there's 2 little ones. We have 2 big chromosomes, 2 little chromosomes. They are genetically identical of what we started with. Okay? Last step, we're going to draw it back in interphase. After cytokinesis has occurred, we now have 2 separate cells. Each cell has the nucleus intact. And our DNA is now uncoiled in chromatin as a bowl of spaghetti. And we started with 1 diploid cell, and we end with how many cells? And are they haploids or diploids?
Diploid.
They are diploid cells. There are 2 big chromosomes. That's a pair, a complete set. There are 2 little chromosomes. That's a pair. That's a complete set. They are diploid cells. Now we call these 2 cells daughter cells to one another. And what we start with is the parent cell. So the parent cell is genetically blank to the daughter cells. Identical, yep. The parent cell is genetically identical to the daughter cell. Mitosis is a process that is done by blank cells. What I'm asking here is like, what type of cells? So I'm going to say we have 2 types of cells in our body. And hopefully I can get us to this point. What's that? No, asexual versus sexual is a form of reproduction. That's not types of cell, that's types of reproduction.
Haploid and diploid.
Haploid and diploid are different types. Yes, that's kind of mitosis happens in diploid cells or creates diploid cells. That's not what I'm looking for here, though. Okay, but yes, there's haploid and diploid. Those are 2 types, absolutely. Those haploid cells we refer to as what kind of cells? Haploid cells are what kind of cells? Egg and sperm are examples of haploid cells. Collectively, we call egg and sperm cells sex cells. Haploid cells are sex cells. These are diploid cells. So if they aren't sex cells, we refer to them as body cells. We call these body cells. Mitosis is a process that is done by body cells. Give me an example of a body cell. What's a cell in you that isn't a sex cell? Muscle cells. Good, that's a body cell. Hair cells, not a sex cell, body cell. Bone cells, blood cells, right? Any other cell besides sex cells are referred to as body cells. Anything you want to see on this picture quick? Any questions you have on this? Okay, then I'm going to ask you to go to Canvas, and under today's agenda, you could go to today's agenda and find the link. Otherwise, under today's agenda, there is an assignment called, Can You Draw Mitosis? The worksheet that we just had was called, Can You Draw Mitosis? I am asking you to take a picture of your drawing that you just did and is now 100% correct because we went through it step by step by step and you corrected yours. I want you to take a picture of that and submit both sides in Canvas because that's a practice to learn activity that you just did. And I want to make sure you get credit for that. We took guided notes on mitosis for a homework assignment last night. I want to make sure that the really, really, really important things that are more than just read some words on a slide, that you understand them, because on Tuesday, December 22nd, we will be testing in this unit on cell cycle, mitosis and meiosis. And some of the information that was in this guided notes is going to be on your test on December 21st or 22nd. Okay? So I want to make sure we are clear. These are the exact slides from your guided notes. I want to make sure it's not just a word that you copied. I want to make sure we explain a little bit more of this. This is the very last slide. I just want to start with that, and then we'll go to the beginning. We are not going to go step by step through meiosis today. We'll do that next week. That's not what I'm going to spend time doing today. All right, so mitosis versus meiosis. We just drew mitosis. We start with one diploid cell. We end up with two genetically identical diploid daughter cells. Mitosis starts with one diploid cell. We end with two diploid cells. We, as humans, have 46 chromosomes. We end up with two cells that have 46 chromosomes. Meiosis is similar in terms of we still have prophase, metaphase, anaphase, telophase. They still occur in that order. It just happens twice. There's something called meiosis 1, meiosis 2. So a diploid cell is what we start with. We go through meiosis 1, and then meiosis 1 is followed by meiosis 2. In meiosis, we start with one diploid cell. We end up with 4 haploid cells. Now, these 4 haploid cells have half the amount of DNA. They have half the number of chromosomes. They are haploid cells. And these produce sex cells. These are sperm cells. These are egg cells. Okay? One diploid cell creates 4 sex cells. That's the big picture of meiosis. Now, when we talk about why things go through the cell cycle, one of the things we talked about was reproduction through the process of asexual reproduction. Differences between sexual and asexual reproduction is something we want you to know. We want you to know the pros and cons of each one. Okay, so asexual reproduction, some organisms, like a single-cell bacteria can go through mitosis where it starts with one cell and in the end it ends up with 2 identical cells as what they started with. If one bacteria cell enters your body, it can replicate and it can reproduce asexually. And given enough time, it can infect you if your immune system can't fight it off. White blood cells, I think it'd have to be. Can't fight off in time, you get a bacterial infection, you might get sick. They are genetically identical to their parents. It's quick, there's no mate necessary. It's a pretty easy thing to do. However, it makes them fairly susceptible to dying. It makes them pretty susceptible to some type of disease. If genetically every single bacteria that is making you sick is susceptible to an antibiotic treatment, if you take antibiotics, you kill the bacteria, that bacteria is now dead in your body, which is good for us, but is bad for them. Right? If there were some genetic variations within that bacterial infection, then you take some antibiotics and you might not be able to kill all those bacteria in you. which is bad for us, but is good for bacteria, right? Survival of that species. And we talk about that a little bit with antibiotic-resistant drugs. That's not necessarily genetic variations of bacteria cells in you. That's in our population. So sometimes we get a bacterial infection that we don't have antibiotics, which is really kind of scary, that we can treat. But through the process of asexual reproduction and mitosis, some single-cell organisms like bacteria can reproduce, and it only takes one. There are other organisms like plants and some fungi that can go through asexual reproduction in a little bit different format. Hanging by the window, I have two plants. One is longer than the other one. Those two plants are genetically identical to one another. Those two plants are clones of one another. The plant that's shorter and is closer to you guys is a product of asexual reproduction. I took some clippings from that long plant, I physically snipped them with a scissors, stuck them in the dirt, and those started to regrow. That's a form of asexual reproduction. They are genetically identical because it was a plant. It's basically like saying if I chop off my arm, that my arm would grow a new me. It's not how we reproduce, but that's essentially what happened with some species like that plant.
So don't try that.
No. Your arm will not regrow either. Not only will your arm not grow into a new you, your arm does not regrow either. So, but in some species it does. Like a starfish, that arm would regrow, and depending on where that arm is severed, that arm might grow a new rest of the body, depending on the species and depending where it is. So there are some organisms that you can just physically like chop in half, and then each half will regrow into a new one, but not all. Like an earthworm. If you cut an earthworm in half, that's not two new organisms. That's one organism that has some of the like digestive parts, the heart up on the top where that band is. The other part doesn't have a circulatory system anymore, but it's wiggling, it's moving because it's slowly dying, but it's still two separate organisms. It's still one organism, just in two pieces. It's not gonna grow again to a new piece. That's an asexual reproduction. Okay, that plant is a product of asexual reproduction, but it wasn't through mitosis. So there's other ways to asexually reproduce without mitosis. But that's kind of what we're talking about. We are a product of sexual reproduction, and in sexual reproduction, two parents are required. Now, when we say two parents, we often think of us. We have a mom and we have a dad. That's why we're here. But you need two parents. Now, some organisms it is possible for some organisms to self-fertilize. They have both male and female sex organs, and then they can self-fertilize. Example, a flower. A flower produces pollen, and it has eggs. Pollen is like the sperm of the flower world. And some flowers, not all flowers, but some flowers can self-pollinate. They're sexually reproducing with themselves. Some organisms can. We can't, but some can. So we just generically say we need two because we do still have a male and a female counterpart that are combining through sexual reproduction. We make sex cells through the process of meiosis, and these sex cells are genetically different from our parents. The huge benefit of sexual reproduction is genetic variation, and that kind of goes along with little genetic variation. It makes organisms susceptible to diseases and extinction. So we'll spend more time talking about that in just a moment. One of the downfalls of sexual reproduction is you need a mate in order to succeed, or for the species to survive. So a lot of times we talk about survival of a species, not survival of you. We talk about the survival of humans, we talk about survival of endangered species. We talk about survival of a population, not individuals. So in order for a species to survive, we often talk about genetic variation is beneficial. But we, as sexually reproducing organisms, we need a mate. Okay, so if something were to happen that you were the last person on Earth, is the human population going to survive? No, right? There is the white rhino. There are 2 or 3. I saw 2 different numbers. It's either 2 or 3 white rhinos left on Earth. back in 2018, they said that, I think the term is they're essentially extinct, but they're dead. They're a dead species. They're not going to make it. And you say, well, how do you know? There's still 2, there's still 3. They're all girls. They are sexually reproducing organisms. There's no mate left on Earth. The last male that we know of, which I'm pretty sure we're probably pretty yes, is gone in 2018. So even though there are 2 or 3 females, as of right now, that population is done. Unless somehow we as scientists can intervene. They've been trying. And they tried before that male died too. That population's done. Okay, so when we talk about survival of a species, that species is done because their mates are gone. Now, if rhinos were asexual reproducing organisms, all you gotta do is chop them in half, and we got 2 rhinos. But that's not how it works.
A
Okay,
little budding,
what's that?
little budding.
Budding, yeah, so budding is a process like if I cut a finger off, kinda, and this is very simplified, but basically if I take a piece of my finger, then we plant it, not really, but and then that finger would regenerate. It's kinda like what I did with the plant. Kinda. Very poor. It was a poor description of it. But that's the idea of that. We are, however, are sexually reproducing organisms. So when we talk about genetic variation in geese, I said genetic variation is one of the major benefits of sexual reproduction. And there's an entire slide talking about genetic variation. And I am talking about genetic variation specifically to you. Probably a good reason for on December 21st and 22nd that we're going to talk about genetic variation. Okay, so when we talk about genetic variation, again, we're talking about survival of a species. So if an organism reproduces asexually, there is very little genetic variation in that population, which means they're very susceptible to some disease wiping it out. There are some species or crops that have been completely wiped out throughout history, I should say. And the reason is they were all basically clones of one another. They had no genetic differences. In agriculture, it's not uncommon that they will take like an apple tree, and they say, hey, this apple tree produces good tasting apples, and they can cut off some branches and they can graft it onto another plant, and then now that tree is now growing a different type of apples. Okay, they do stuff like that. Or they say, hey, this is a good one, we're just gonna plant it again, we're gonna plant it again, and we're gonna plant it again. There are variations of corn, right? You can go to the farmer's market and you see different variations of corn. But some crops, there's like one variation of. Bananas are a great example of something that they call a monoculture. There's basically one line of bananas that is developed and harvested and sent throughout the world. And if some disease were to come through, can plants get diseases? Absolutely. Okay, and a plant can get a disease, and if some disease came through and said, hey, I like that type of banana plant, it could completely wipe out all banana plants as we know it. And then, in essence, the banana plant could go extinct. Now, there are some other variations of bananas throughout the world, but in terms of our agricultural, what we harvest for food, there's basically one line. And that's happened in the past. Banana crops have crashed because we had a monoculture. A disease came through and wiped it out. So what did we do? We redeveloped a different monoculture of a different one that we still are banking all of our agriculture on. So it could happen yet again. If we don't have genetic variation within a population, if something comes through, it's pretty likely it's going to completely wipe out that thing. Same thing with us, right? If we had some disease that spread throughout the world that was very infectious, we as a human population could become extinct. Now, I'm not saying COVID is that. Are people dying from COVID? Yes. Are people recovering from COVID? Yes. Are we trying to develop a vaccination to help people from getting COVID? Yes, absolutely, all this stuff. But in history, have there been infectious diseases that have spread throughout the world that have been really, really, really deadly?
Yes.
Yes. Example?
Black Death.
Yeah, the Black Death, the Black Plague, the bubonic plague. Okay, that was really, really bad. I'm not trying to downplay COVID. Please don't take that. But Black Plague was bad. If you got sick, you pretty much died. And if you got sick, they would quarantine you in your house with your family, and it's very infectious.
Guess what happened to them?
They got sick, and then they died, and entire villages were dying. Okay, it was really bad, right? If you look at a world population, you see a dip in the world population during that era. So it was really bad. It was very infectious. However, if we were all genetically identical, then as a population, we would be susceptible, very susceptible to extinction. But we have genetic variation in our populations, which makes it better for us for survival of a species. We are more likely to survive a Black Death, a Black Plague outbreak because we have genetic variation in our population. We have these things called mutations. Sometimes they're harmful, but sometimes they're beneficial. If it's a beneficial mutation, for example, I have some weird mutation in my body that the only thing it does is prevents me from getting that disease. Hey, that's a good one. That was a good one for me to have. And as long as there's more than one of me in the world, our population could live on. Survival of a species, not an individual, survival of a species. Okay? So some of the times we have these genetic variations that allow us to be resistant to these things, naturally resistant without a vaccination. And there's sci-fi movies, there's movies about outbreaks of things that people have these immunities to, and it's true, it's a real thing. During the Black Plague, there were some villages in Europe that they can trace ancestry, and there was a higher survival rate in those villages because, and they can now look at modern genetics of lineages, family lineages, and they say, hey, there's something different right here on their DNA. What is that? And the same thing is true with the HIV virus. There are some people who just don't get HIV because they have some mutation on their DNA, in their chromosomes. So survival of a species is helpful for survival, or genetic variation is helpful for survival of a species as a population. And we can get these variations through sexual reproduction. I want to make this point. Internal fertilization is not required for sexual reproduction. I'm saying, by lack of better terms, sex isn't required for organisms to sexually reproduce. There are some organisms who sexually reproduce, a combination of egg and sperm, without actually going through the act of sexual intercourse. Case in point, a fish. They fertilize their eggs externally.
Still sexual reproduction.
Egg and sperm combining. It just happens.
not-
It doesn't
it's not
happen
the
in the
process.
body, right? It happens in the bottom of the lake, the bottom of the river. Flowers, same thing. Pollination, that's sexual reproduction. Sexual reproduction is not, internal fertilization does not require, internal fertilization is not required for sexual reproduction. So we talk about chromosome numbers a lot. We talk about us having 46 chromosomes. So if we we talk about diploid numbers and we talk about haploid numbers. Our diploid number is 46. What's our haploid number? Well, haploid is half of that, so 46, we have 23. Now, we can expect questions like this. Hey, a fruit fly has a diploid number of 8. What's its haploid number? Half, it's 4, right? Or we can go the other way and we can say, hey, chimpanzees have a haploid number of 24. What's their diploid number? Well, it's double, right? Because this is half, so what's 2 of those? Double that, it's 48. Okay, haploid, diploid numbers. We get genetic variation within our populations because of three things. This talks about 2 of the 3. One of the things is random fertilization. When we talk about random fertilization, what that's referring to is like these fish eggs that are laid in the bottom of a river are going to be randomly fertilized by some random sperm. We don't know what sperm from this male fish is going to fertilize that egg. There's millions of them. There's lots of eggs. Which one is going to fertilize? We don't know. That's referred to as random fertilization. However, when we make sex cells, there's 2 different reasons why we get genetic variation within our sex cells. They're called independent assortment and crossing over. During meiosis, we're going to talk about this on Tuesday. During meiosis, we have these chromosomes. These are homologous chromosomes. One's from mom, one's from dad. This one happened to line up blue-red. But it's random, it's a flip of a coin. We don't know if it's going to go blue-red or red-blue. Independent assortment says that these 2 lining up like this are independent of these 2 lining up like this. It's a flip of a coin, whether it goes heads, tails is independent of whether it goes tails, heads, tails. It could be this, or it could be this. Every time those chromosomes line up in the middle, it's random, it's independent of the previous time. Now what's gonna happen is these homologous chromosomes are gonna separate, these homologous chromosomes are gonna separate, and if this was a sperm cell, and this was a sperm cell, and this was a sperm cell, and this is a sperm cell, these four are all genetically different from one another because of how these chromosomes lined up here. The other way that we can get genetic variation within sex cells is something called crossing over. We have 4 sister chromatids in this picture right here. 2 homologous pairs. What's true about our sister chromatids? They're the same, they're genetically identical. However, during prophase 1, we get these things called tetrads where these 4 are 1, 2, 3, 4, they're really close to one another. We can get a swapping of genes. And what happens is part of this blue chromatid attaches to the red, and this part of the red attaches to the blue. Now, these 2 sister chromatids were genetically identical. Are the 2 sister chromatids still genetically identical? There's variation. Now, through the process of meiosis, this sister chromatid is going to go into one sperm cell, or sex cell, I should say. This is going to go into a different sex cell. This is going to go into a different sex cell. Same thing with this. We will form 4 sex cells. All 4 of those sex cells are now genetically different because of that crossing over event. There's genetic variation there. There's also probably genetic variation because of independent assortment. So we get lots of genetic variation when we go through and we make sex cells. Now, all of these things we're going to go through is ultimately making sex cells. Meiosis forms these things called gametes, which are sex cells, an egg cell or a sperm cell. It's the technical term to say a haploid cell, or a sex cell. It's an egg or it's a sperm. 23 chromosomes, 23 chromosomes. Through sexual reproduction, we restore the diploid number. We take 23 plus 23, we get 46 chromosomes in a fertilized egg. 46 chromosomes. We restore the diploid number. The diploid number is restored. The fancy term for a fertilized egg is called a zygote. But then that zygote now has 46 chromosomes. It's a diploid cell. It goes through mitosis and mitosis and mitosis, develops into embryo, eventually you today because of that. There are some variations that can happen when we have fertilization. Most of the time, one egg is fertilized by one sperm and it develops into one person or one organism. But sometimes that's not the case. We get multiples, or we get twins, or triplets, or quads, or whatever, but we're just going to talk about twins. There's 2 basic types of twins, and the first is identical. Identical twins is when one egg was fertilized by one sperm. 46 chromosomes. 23 plus 23 is 46. What happens somewhere early is that fertilized egg split into 2 separate things, and then those separate fertilized eggs developed into 2 individuals. One egg, one sperm fertilized with 46 chromosomes. That one cell went through mitosis and divided in half. Those then went through mitosis. Genetically, that one egg, that one sperm that were fertilized are genetically identical if we're identical twins. They are clones of one another. Their DNA is exactly the same because it was one sperm and one egg that was fertilized to make one individual. Because they are genetically identical, they must be the same gender. They must be the same gender because genes, gender based upon chromosomes is differences in the X and Y chromosomes. Okay, so if they're genetically identical, they have to be the same because they are genetically identical and DNA is influenced by the X and Y chromosomes. The other type of twins is referred to as a fraternal twin. In a fraternal twin, it was 2 different eggs were fertilized by 2 different sperm to make 2 different zygotes, which then developed into 2 different people or 2 different organisms. They are brothers, they are sisters of each other. 2 different eggs, 2 different sperm. They could be different genders or they could be the same gender because there's 2 separate eggs that were fertilized. Anybody in here twin? No twins?
Is this the first?
I think this is the first
The
class.
first class?
Yeah. I have twins. I have a boy-girl set of twins, Will and Eve.
Well, so fraternal, or identical? Hmm?
Right? My kids are not identical to one another. One's a boy, one's a girl. They are a different gender. However, you could be fraternal twins that are the same gender. Boy, boy twins, girl, girl twins. The fact that mine are boy, girl, they must be fraternal. Even if they look similar, they're still genetically different. 2 eggs, 2 sperm, they're genetically different. The fact that my kids were born 1 minute apart, they had the same genetic similarities that they would have been if they were born 2 years apart. It was 2 eggs, 2 sperm, fertilized, just at, they were born at the same time instead of years apart. That's it. Okay? So why do they look so much alike when they're babies? Because they're the same age with the same genetic information. Just like if you look at baby pictures from when you were 1 and your brother or sister when they were 1, probably look pretty similar to each other at age 1. They just happen to be the same age at the same time and right next to each other at every given moment. Well, when they were young. Okay, so that's the difference between twins. So those are kind of the key points that we're hitting. Whew, I timed up pretty well.
The board here as well. You can take a picture, otherwise you guys know. For anybody that did not pick this work of art up, this was the Pogo on Tuesday. This was the Pogo on Tuesday where we gave it to you a little bit early because it is a little bit longer. If you didn't pick this up, there's copies right here in the blue bin. If you did, that's your assignment moving forward.
We'll draw meiosis on Tuesday or Monday or Tuesday.